Hi everyone, welcome back to CHM421, which is analytical chemistry. This time we will continue our lecture on topic 3, which is sampling and evaluation of experimental data. As usual, this is the description of this subject. It involves the theories and technique in analytical chemistry. Student will define the concept, explain the principle and application, solve qualitative and quantitative problems, perform investigation via laboratory exercise. The outcome should be assessed through paper examination, test and quizzes, laboratory scale, and also laboratory reports. The course learning outcome, students should be able to describe the function of laboratory apparatus, standard laboratory techniques, as well as the principle that we use in sampling and preparing the laboratory samples. Explain the principle and application of volumetric, gravimetric, and basic chromatography analysis to solve the reliability of experimental data. To demonstrate the theories that covered by performing the experiments on volumetric titration, gravimetric analysis, and chromatography. At the end of the course, a student also expected to be able to write experimental finding in the scientific manner. For topic 3, is quite a long list of outline. We will divide this topic 3 into 3 subcategory. Uh, First, we will cover about sampling. It consists of sampling methods, how to reduce the sample into laboratory size sample, uh, how to do the dissolution of sample before we do the analysis, and also the eliminate any interference in the samples. The subcategory number 2, we evaluate the experimental data. In the subtopic of evaluation of experimental data, we will learn a little bit on type of error, mean and median, and also the precise and accuracy of the data that we collected after do the analysis, and also do the standard deviation in all standard deviation. The last subtopic under the this topic 3 is more on to statistical. We will learn the related statistical technique to evaluate the experimental data that we collected in the lab. So in this subject, we will learn a little bit on the confidence limits and significant tests, gross error, and also Q-test. So for this video, I will talk about on the subtopic of sampling, particularly on the sampling method. There are several analyses in research or in the laboratory, like we can do the analysis for physical and chemical properties. We can do the qualitative analysis, quantitative analysis, and also diagnostic analysis. However, in this subject, CHM421, we will focus more on the qualitative and also quantitative analysis. As we learned in the previous topic, we covered a little bit about what is qualitative and what is quantitative analysis. As you remember, the qualitative is about we want to know the chemical identity of the analytes present in the samples. However, for the quantitative, it's more to about amount. We analyze or we determine what is the amount, meaning that how much uh, in terms of concentration or in terms of mass of the analyte in the sample that we analyze in the lab. So this is the general steps involving in any analysis, uh, include in analytical analysis. First, before we do variable analysis, we should identify what is the problem that we want to solve. That's number one. After we determine the problems, and then we design the experimental procedure. Uh, During designing experimental procedure, there are several issues or several things that we have to consider. The first or the most important part is uh, select the suitable method, meaning that the sampling method should be correct to ensure the result that we obtain from the analysis precise and accurate and also correct. After we design the experimental procedure, after we confirm, and then we proceed with conducting experiment. What is the purpose of conducting experiment? We want to gather the data. After we obtain the data from the analysis, we're gonna to analyze it. We're gonna to evaluate the data. It's the data acceptable, reliable? Is it the data accurate, precise? So during the data analysis, we will do the statistical, we do verification of result, and also we have to interpret the results. And last but not least, we will propose the solution of the problem that we identified in the beginning. So this is the whole circle of the analysis. So, to be specific for step in quantitative analysis, we want to determine the amount, how much the analyte in the sample. This is the step. So, it's about similar with on the previous slide. First, of course, as I told you, we have to select the correct method. Second, after we have the method, and then we have to obtain the representative sample. meaning that you have to collect, you have to do the sampling in order to get the sample to analyze. And then we have to prepare the sample into the lab size sample. So this one we will talk later. Uh, After we do the right size of sample for analysis, we have to do the sample replication. We have to replicate the sample in order to produce many results. Uh, and then we perform decomposition in dissolving the sample. We eliminate the interference. We measure the properties of the analyte that we interested, and we calculate the results. And last but not least, we use the statistical technique to evaluate the reliability of the result. Moving on the uh, method selecting. Okay, several issue we have to think about when we consider either we want to use a method A, method B, or method C, etc. etc. We have to think in terms of accuracy, meaning that the result that we obtain later, how accurate it is. So if you want the level of accuracy, if you want a higher accuracy, normally higher accuracy method, it will be time consuming and it will be a bit costly. And also we can, if we think about the accuracy, so meaning that we have to replicate the samples on several numbers. So for obtain higher accuracy of result, we have to do many replication. And samples that are complex sample can be very simple sample, or sample can be very complex. So the more complex of the sample, it will be increase the costing of the analysis. So when we do the selection, we have to compromise between the accuracy and also economics. Of course, accuracy is number one to tell the reliability of the result that we obtain. At the same time, we have to consider in terms of cost. Of course, we want the analysis cheaper. Okay. All right. So now we focus on the sampling method. Before we go a bit detail on the sampling methods or sampling techniques, we have to understand several things. First, population. What is population? Population is the group of things, items, or unit under the investigation, meaning that population is a large area. And then from the population, we collect the sample. What is the sample? Sample meaning that we obtain the collecting information only about some of the population. So if this is the population, so one of it we take it is a sample. And then after we collect the sample from the population scope of investigation, we can proceed with sampling technique. So sampling is a process of obtaining a small mass of material where the composition accurately represents the whole bulk of population. All right. So three step in sampling. Of course, before we do the sampling, we have to identify the population, and then we collect the gross sample, and then the gross sample have to reduce into the lab sample. Uh, Before sampling, we have to consider the sampling instrument, uh, the uh, container, the sample container. We have to consider the person do the sample, in terms of safety, the sample information, and also how to store the sample storage. All right. What is the purpose of sampling? Uh, the purpose of sampling is to obtain the representative sample. As I mentioned, we want to get the sample representative of the whole population that can be taken into the laboratory for chemical analysis, and the results that we obtain will be accurate. Uh, Normally, during the chemical analysis, only small portion of material is needed for the chemical analysis. So, if the amount of material or amount of samples is very small, and then not needed for any further use, we can use the entire sample to for the analysis. Okay, so sample can be categorized into three. We can do the sampling for gas sample, sampling for the liquid sample, and also sampling the solid sample. So in this video, we will learn how to do how to select the right method to do the sampling for different types of sample. So first is we do the gas sampling. So the technique that we use for the gas sampling, it can be used grab sampling, or we can use continuous sampling. So for the grab sampling, normally we will taken the actual sample A in the sample equipment or sample device. So the actual sample A can be put in the gas sample bag, for example, Teflon bag, or the actual sample A can be put in a flask, or a bottle, or any other suitable container to store the actual sample of A. So after we collect the A sample by using a grab sampling, we are sent to the laboratory to do the analysis. Okay, continuous gas sampling. In continuous gas sampling is a little bit different from the grab sampling, because in continuous, the gas or the vapor should be removed from the air by absorb in the solid or liquid solvent. So as you can see in the grab sampling, we taken the actual sample of A. put it in the container and then send it into the analysis. For the continuous, the A that we focus on, the gas or the vapor in the A should be removed. So how it removed? So the gas in the vapor should be passed through the solid or liquid solvent. So we have several example of solvent for the continuous gas sampling. First, we are using the this sample pump. Okay, so a sample is collected by opening a tube, connecting it into the sample pump. So the A will be pulling in through the tube with the pump. And then the gas or the vapor, the airborne of the chemicals in the A will be trapped in the surface of solvent. So in this case, the solvent, the solid solvent is here. So this is the example of solid solvent. So this tube normally is the it have a cap. So after we collect the A sample, and then we dismantle, and then we cap it the solvent, and then we send into the lab. So this we can have several number of solvent tube that containing the A, the gas or the vapor of the sample that we are interested to analyze. So this is the detail inside the solid solvent for the continuous gas sampling. But you no need to remember all these components as long as you know during the continuous sampling, you will use a solvent tube to do the sampling. So this is the scenario when the worker are wearing a solvent tube and sample pump to determine his exposure to the chemical hazards during the entire work of shift. So as you can see, this is the sampling pump, the sample pump. So wherever A surround him, A will be fallen in, go through the tubing tube and absorb into the solid solvent. So if you want to the content of the A, dismantle this solid solvent and then submit into the lab for the analysis. Okay, all right. So another way if to do the A sample, A sampling is using the rotary vent pump. So this one is normally if you want to know the A surrounding inside a particular room or a particular area, we can using rotary vent pump. Okay, we done the gas sampling. We have grab and continuous sampling, either using the put the sample in the container atau or using the solvent for the continuous sampling or using the pump. So now we focus on the liquid sampling. Uh, as we know, any liquid sample normally it is homogeneous. So meaning that it is more easier to do the liquid sampling. So by shaking the liquid, we can homogenize the whole liquid uh, sample. So let's say we want to do the sampling of the water from the river. So the sample have to be collected in three, at least at three different position or at three different location, on the surface, uh, in the middle, and also in the bottom of the river bed. Uh, or if we have a large container of liquid that we obtain from the uh, sampling site or from the population, so we have to homogenize the liquid in the large container by stirring, and then also we have to take the sample for analysis at three different locations, on the top, middle, and also bottom of the container. All right, this figure is showing the researcher do the liquid sampling, uh, water water sample. So the first picture on the top left is do the sampling water in the surface. He just use plastic bottle to collect the water sample. In this lady using special equipment because she want to do the sampling of the liquid, which is water, at different depth of river. As I told you, we have to collect the sample on the top, in the middle, and also in the bottom of the population. In this case, I think this is a lake. So this special equipment, we call it vertical water sample that have rope uh, that we can adjust the distance during the sample collection. After we collect the liquid sample, we have to transfer the liquid sample into the storage bottle. So as you can see, this is the vertical water sample. We put it into the sample storage bottle. Okay. Uh, all right. This is same thing. Is showing the diagram of researcher do the water sampling using the vertical water sampler, or even we can use plastic bottle, test tube, uh, and also plastic bottle. But using the plastic bottle or test tube, we just can do on the sample collecting on the surface only. All right. Okay. So we done the sampling for gas and also sampling for liquid. Now we focus a little bit on the sampling for the solid. It's quite different from the liquid sample. Liquid sample is homogeneous mixture, but solid sample normally is in homogeneity, because in the solid sample, it can be varies in terms of sample size and particle size. So it is a bit difficult to do the sampling is compared to the gas sampling and also liquid sampling. So of course, the easiest way to do the solid sample is by using the graph technique. However, even though it is easier, but the result is unreliable because when we use the grape sampling for the solid sampling, uh, we just taken the sample at random and we assume the sample that we collect is representative for the whole population. So normally the solid sample have to be for the treatment. So for example, if we have ores that being sampled, so the ore have to be crushed into the smaller size, and then we save it, and then we use quartering technique. So this is technique very important for the solid sampling. This quartering technique is a technique to get the right sample to the laboratory. And maybe solid sample that we collect from the population have to do dry. All right. What is quartering technique? Remember, quartering technique is the right technique to do the solid sample to get the right laboratory sample size. So, quartering technique normally, if we have a sample, we divided it into four sections like this. uh, two diagonal section have to be discarded, and the another two have to be retained, and then we mix it again. So this diagonal section we have to remove, and this the another two we have to retain, and then we mix it again. And then after we mix, we redivided into four section. We repeat. The two diagonal will be removed and another two will be written and mixed. So this process of removal and remixing will be remain until we obtain the correct amount of sample for the analysis. And then it can be this technique can be carried out with ranging of sample. A small sample also can using filtering technique. Very large sample also can be used too. So just remember, for the solid sample we have to apply the watering technique. Okay, so this is various example of solid sample. Of course, we can have paper, we can have glass, plastic, sometimes e-waste, electronic waste, metal, or agriculture or organic waste is the solid sample. As you can see on the top, on the left is the soil sample. Uh, Vegetable also can be is a solid sample. Fish also we can do the sample solid sample analysis. So how to collect the sample? Uh, just now we learned about the quartering technique. Quartering technique is to get the right sample size for the laboratory. But before we apply the quartering technique, we have to collect the sample. So how to collect the sample? So if it is solid in the loose form, So we can use the conveyor, or we can use the teeth. Either use a split tube teeth or concentric teeth if the samples is loose powder. If we have a very hard solid sample or very compact solid sample like metals, rocks, clay, so we have to use the right tools in order to collect the sample. So in this case, we can have we can use a teeth, or we can use auger. For the metal in alloy, we can do the drilling, or we can do the milling in order to break the sample particle into the small size. So remember, don't just taken the sample by scraping. Okay, so we have to do the drilling or the milling. So this is a several example of tools or apparatus to do the solid sample. We can use the tablet sampler, or we can use a scopper, or we can use the in-depth solid sample. Okay. So this is several example of auger. Auger is for the drilling device. So it can be come different shape of auger. Uh, it can be post hole auger, or it can be helical, or we can use extension root, or what else? Gravel auger, uh, open in close a spiral, or a flat spiral barrel auger. So several many types of tools or apparatus available for the soil sampling. Okay, so I think that's it for this video for the method sampling. At least you learn how to do the sampling for gas, how to do the sampling for liquid, and also how to do the sampling for the soil. So on the next video, we will continue on how to reduce the sample we collect from the population or the sampling site. We name it gross sample into the laboratory size sample. So see you on the next video. Okay, bye bye. Thank you and bye bye.